Good evening, everyone. You are welcome to another opportunity at Edu Interact AI. This is going to be another time, and today we are going to consider Krebs Cycle, Glycolysis, and fermentation. So, you don't need to cram anything. What you need to know is to be able to know how to hack and the shortest way be able to remember these three very important journey in metabolic pathway. And today, just like I said, there's going to be both post test as well as a pre-test. So let us just kick off with the infographic for today. Infographic for today. As it is being displayed, you will see it as it is being displayed. Let us try as much as possible. Let me try and give you some hints, and thereafter we'll be able to look at how this can be. So later on, we will be able to. I will give you time, then I will give you the quiz. Don't forget that the quiz should be submitted at the comment section. So you submit only the correct answer. Submit it at the comment section. Now, as you have it here displayed for you, this is termed the energy extraction toolkit. That is a simple guide to cellular pathway. I want you to imagine and I want you to remember the correct structure of a cell, a living cell. When you have a living cell, the cell is in the center of any living cell, especially eukaryotic cell. In the center of eukaryotic cell, you will have what is called the nucleus. The nucleus. So a cell is basically divided into two types. Sorry, into two parts. The first part is called the cytosol. Cyto. Anywhere you come across the word cyto, cyto is telling you about cell. Cyto. And in some textbook, you will see it, the cytosol is also referred to as the cytoplasm. Cyto, that is the cell, and plasm means the liquid. So the blood of the cell, the liquid parts or the liquid contents of the cell, like water, like water, is called the cytoplasm. So the cytoplasm is like the water in which organelles will swim. The organelle, imagine it as the fish in the ocean. So the water is the cytosol. Now, leaving what is called the cytosol, there are some organelles, small organelles, small organs within the cell. They are called these organelles. So organelles are small membrane bounded organelle, that is membrane bounded parts. Just like in the whole of human body, you also have some vital organs like the heart, the liver, the kidney, and so on. Inside the cell, you also have all these small organs that are called organelles, like the Golgi apparatus, mitochondria, the endoplasmic reticulum, lysosomes, ribosomes, cytoskeleton, and so on and so forth. All these are known and are called small organs because sometimes ago you will remember the theory of endosymbiosis. When we say endo in biology, what it means is inside, and symbiosis is the mutual relationship between two partners, mutualistic agreement, that is based on agreement, certain agreement. So what we are saying in essence is all these organelles, they are to certain level independent. Independent because they are being independent because they have their own function that they play. They have their own role that they play. So what we are trying to tell you is just that organelles are small membrane bounded parts that are embedded or that are within the cell. So when we are talking of what is cellular respiration, if we are talking about what is glycolysis, and if you are talking about anaerobic or fermentation, that means we'll be talking about what takes place in the cytoplasm. That is glycolysis. Take place in the cytoplasm, then it will now move from the cytoplasm down to the cristae, that is in the mitochondria. Now, what we are trying to tell you is that don't forget that any business at all that is not going to fetch you money, any business within the cell that is not going to fetch you the energy currency, which is called the adenosine triphosphate. I told you the last lecture that ATP are just the naira of the cell currency. If there's going to be any cellular activities, involvement of ATP is very, very important. Any business that does not actually involve this money, either expending money uh, or even making money as a profit, that is no longer a business. That is another thing entirely, and nobody is going to show it. The same thing within the cell. The mitochondria within the cell is called the powerhouse of the cell. Why is it called the powerhouse of the cell? Is because that is where the uh, ATP, that is the cellular currency, is being produced. So the mitochondria can also be regarded as the energy center, as the central bank of Nigeria, where money is being produced or is being uh, printed. So that is mitochondria. So don't forget that. So that means if mitochondria is the center of ATP generation, how how is this ATP being generated? In the last lecture, I actually taught you about how can you compare these three types of cellular respiration. Uh, talk about glycolysis, how much money in form of ATP is being invested, and how much money is being realized in form of ATP. We were able to look at glycolysis as a kind of business in which 4 ATP is being realized. And we also look at fermentation. Fermentation, where don't forget that glycolysis, in glycolysis, at the end of the day, you have pyruvate acid or pyruvate or pyruvic acid is the three carbon compound that is being generated. So some of us will find it very, very difficult and too cumbersome, especially when it comes to if a question or certain question is being asked for you to be able to talk about all these pathways. You just discover that you might just be scratching your head without even knowing how to say it. So this is our business today is not to memorize anything. It's just for you to be able to discover the secrets of how can you, as a student, how can you monitor, how can you understand without even memorizing anything? What are the shortest form, the shortest pathway uh, to this all these pathways? And that is what is showing to you here, where you have what is called the energy extraction toolkit, which is a simple guide to cellular pathways. Now, as you can see it from here, that we are talking about glycolysis. Glycolysis, you can be regarded and can be known as spend, split, and sequence. You spend, split, and sequence. Now, in first 2 ATP, the cell is going to invest 2 adenosine triphosphates, and it is this 2 ATP that is going to activate the glucose, and the glucose is going to be splitted. That is, don't forget, glucose is a 6-carbon compound. So whenever it is, you can see it here, 1, 2, 3. So this one will be splitted into 2. That is when ATP will be infested. And once it is being infected, I want you to remember, that um, PFK is going to be splitted into 2. There will be a part. If you look at this place, you will see that 6-carbon compound will be splitted into 3-carbon compound, which is called the pyruvate. So when it gets to the level of pyruvate, pyruvate now has 2 options. It is either it goes into fermentation, and if it is going into fermentation, that is to tell you that the electron acceptor is going to be an organic compound, and that organic compound is going to be ethanol. So that one can be recycled back into what is called glycolysis, and glycolysis will just pick up again, and it will lead into fermentation. But don't forget that all these that are involved, all these reactions, biochemical reactions, are taking place without oxygen. And any oxygen, any pathway that does not, especially when it comes to respiration, any pathway that does not involve oxygen as the electron acceptor, that means it's going to be in deficit. The cell will not want to embark on that kind of journey. So just like I told you in the last lecture, that everybody wants to get involved in any business that is going to bring return on investment, high return of investment. So pyruvate, the pyruvate can proceed via what is called fermentation. That is why fermentation is a recycling service. It does not make extra ATP. What it does is just to restore NAD plus to keep the um, glycolytic reaction, to keep it from running around. Okay, now the second alternative, apart from if the cell is going to proceed on fermentation, the second alternative is going to be when the pyruvate acid is going to undergo several other oxidative reaction that is going to involve oxygen taking place. And at that point, hello, okay, so and at that point, you just discover that it is going to proceed on Krebs cycle. It is going to proceed on Krebs cycle. And any reaction that is going to be on Krebs cycle, that means it is going to be what? is going to involve oxygen. So without wasting our time, now you know that the whole process, whichever way the cell wants to go, if the cell is going, so the whole process is going to start with glycolysis, and at a junction, the glycolysis, the glucose will be splitted into two carbon compound, I mean two molecules of pyruvate, that is the three carbon compound. It will be splitted. So that one we have one or two of these alternatives. The first one is that it will proceed via what is called fermentation, and thereafter the fermentation will now restore energy back so that the process of glycolysis will be able to start all over again. So fermentation is a recycling service. It's a way in which the cell is going to return to its original state before any. Yeah. So, and these two phases will proceed without oxygen. Fermentation, that means the electron acceptor is going to be an organic acid. Then glucose, that of glucose, that I mean glycolysis, that means it is going to proceed via nitrates, sulfate, and so on. Okay, then the next one where oxygen will be very, very active is called the citric acid cycle, or what is called the Krebs cycle, so to say. And if you look at what is happening here in terms of energy generation, so you see this is cytosol. Cytosol is the same thing as the cytoplasm. Now, this is where you have the pyruvate, and you have two molecules of this pyruvate. So the pyruvate must lose a carbon and capture what is called the electron before it will be able to enter straight into the crystal of mitochondria. So, and that one is going to proceed via the Krebs cycle, or what is called the citric acid cycle, in which when you invest only 2 ATP, you are going to get nothing less than 32 in returns, depending on the prevailing environmental condition of that cell. So this is the situation in which we are. Now, I want you to take your time now in the next few minutes, especially let me just say 10 minutes. We are going to do this pretest, and that's going to be what we are going to start with. So I only wanted you to understand the basic principle of this. Okay, so let me try and display this to you so that you'll be able to do it. Just write the correct answer. Don't need to write too much. Just write the correct answer. Is a multi-choice question that is objective. Just pick the correct answer and let us see how it is going to go. Okay, so this is number one. Okay, So it is going to be, this is question 6. I'm going to be faster now because we still have 15 minutes for the presentation. 15 minutes for presentation. Okay. How many carbon atoms are released as CO2 during the break step of pyruvate oxidation per molecules of glucose? Okay. Yeah, I won't display the correct answers until later on because of some people are fond of waiting for the answer before they will be able to submit, and that would not in any way um, be right. Okay. Don't forget the essence of pre-test. We want to test your knowledge before the presentation, before the real class. So even if you score 0 in the first presentation, but after you must have listened to the lecture, we want to see that is post test is very, very important. Post is very, very important. Okay. So why is Krebs cycle described as cycle rather than a linear pathway? So this one is calling for your thoughts. Okay. Glycolysis is always almost like a straight line from step 1 down to step 10. However, the Krebs cycle that's just about 5 or 6 molecules. They are a form of cycle. So why? So you need to respond to that. Okay. So for the pre-test, I'm going to allow only 10 questions. The post is going to be the one that is going to be 15. So that is telling us that this is going to be the last question in the pre-test. In the pre-test, this is going to be the last question. What is the net gain of ATP per molecule of glucose if the cell is limited to fermentation? Okay. So it is on that note I want to see your response in the comment section. I want to see your response in the comment section. Okay. So let's go on with the normal presentation. So that is hacking cellular metabolism. Hacking cellular metabolism, and that's going to be this presentation. So I want you to imagine, look at this diagram, the biological pathway toolkit. So you have to stop memorizing those reactions and you have to start tracking exactly what happened. It's not a matter of you should memorize. No, you should not. Looking at this system, you will see anything that has to do that is occurring in blue, that is carbon flow and molecules. And as you can see it here in this cellular machine, you see it here. All these ones that are in blue color, that is telling us that that is carbon flow and molecular workflow. Okay. The next one that is going to be in orange, that is electron NADH and FAD2. Okay, so then whereas the main one, the green, these are uh, points where ATP are being generated. So you can see that ATP is being generated inside and inside the mitochondria. Now the question now I want to ask, the puzzle I want to ask you, and I'm expecting your response in the comment section, is that we are talking of oxidative, or let me say aerobic respiration. Aerobic respiration. And when you look at the structure of microbial cell, a typical microbial bacterial cell is a prokaryotic cell. And don't forget that in prokaryotic cell, you do not have distinguished or noticeable, you don't have noticeable organelle. That is, what you will only see is the DNA, the genetic material. What you will see, you won't see Golgi apparatus, you will not see endoplasmic reticulum. You might see ribosomes, but they are not membrane bounded. They are not enclosed. There's nothing you can call the nucleus in prokaryotic cell. But here, I'm demonstrating to you that glycolysis will take place in the cytosol and aerobic respiration will take place in the cristae, that is inside the mitochondria. So the question is, where will the prokaryotic cell, like typical bacterial cell, where do they get where will their own anaerobic or pathways, biochemical pathways, where is it going to take place? Okay. Now, going through this, I've said in the preamble I gave to you that there are three basic types of respiration. The first one is anaerobic, which is also known as glycolytic pathway. That is where 6 carbon compound will be systematically broken down into 2 carbon compounds of a 3 carbon compound, which is called the pyruvate. You have 2 molecules of it, and each molecules of this, they have 2 fates. Number one is to proceed into fermentation, where the ATP or the pyruvate is going to be recycled back into glucose, where the whole process of glycolysis will take place. Then the second one is going to be through the pyruvate, that it will be systematically oxidized into CO2 and water. And that's going to be the end product of any cellular respiration. And it is going to proceed via what is called the aerobic respiration, or the Krebs cycle. Okay, now I want you to know very well that every pathway, every major pathway that occurs anywhere, number one, you should divide it into 3 aspects or 3 parts. The first one, or the first layer, is going to be the story. The second layer is going to be the ledger, and the third layer is going to be the control points. The layer, which is going to be the story, will be the one that will define the specific biological problem. That is the layer. There must be the need. If the cell is going to proceed on any type of respiration at all, it must be based on the need of that cell. If there is no need to proceed into cellular respiration, the cell will not do that, irrespective of whether it is necessary but not important. So that is layer one. That is the story, which defines the specific biological problem, the pathway solved, and the strategic logic behind it. That is the rationale. The cell will not do anything at all, no matter how. That's why you will never define a cell as just a single independent, a single entity that is just the smallest part. You don't define a cell like that. What you say, whatever definition you are going to give to a cell, you must not forget to put that it is it is capable of independent existence. So what we are saying in essence is that you just consider a cell like an organism, the whole organism. Cell go hungry, the organism go hungry. So if there's no need for you to eat, must you eat and even enjoy yourself? Must you eat and it become a poison to you? So that is the story, that for every pathway there must be the story. The second aspect is that the layer two is the ledger stage. The ledger stage, that is the ledger stage that will track the exact input and output um through using a kind of currency code in form of ATP, or it could be in form of GTP. So what we are saying in essence is that is the ledger. You know, those of us that are very familiar with the accounting, you know what a ledger is. So that is, you'll be talking of your balance sheet. You'll be talking about okay, this is the expenditure side, and this is going to be what? Is going to be the expenditure side is going to be the income side as well. So, and when you want to balance your ledger, that means you must subtract your income from your expenses. That's going to give you how profitable the return on investment that your business going to be. And I've just told you that in the area of glycolysis, the glycolytic pathway is going to invest 2 molecules of ATP and get 4 molecules of ATP at the that at least you invest 2 ATP, you invest 2 naira, and at the end of the day you get 4 naira back. So when you now subtract your income from your capital, or when you subtract your capital from your income, so that's going to give you a net balance of 2 ATP, and that's why that is the ledger stage of any pathway. So you don't need to memorize anything. Just know that any pathway, biological pathway, you must divide it into three aspects: story, the ledger, and the control points. Now, the control points will identify a few irreversible. That is, there are some that once you have made the profit, there's nothing like warranty whereby you need to go back and say, okay, you are not. So the cell will not say they are no longer interested in that business again. So that is the control point. So if the cell is going to be interested, that means more energy must be generated. And if the cell is not going to be involved, at the end of the day, that means energy will in any way be regulated to take place. Okay, so that means 3 layers are involved. The first one is called the story, the second one is called the ledger stage, and the third one is called the control point. Okay, now having that in your mind, now you can see that everything that has to do with respiration, cellular respiration, is going to involve 2 organelles or 2 locations. The first one is in the cytosol, then the zone 2 is going to be in the mitochondria. Looking at what you have in the left, you will see that this is the machine, glycolytic machine, where you extract very quick cash, extract energy quickly, no direct need of oxygen. So that is a glycolytic pathway. So it is going to enter through here. The glucose will enter. It's going to serve as the raw material. And when it serves as raw material, the first step is going to be conversion of that glucose to glucose 6. Glucose 6. So immediately after the conversion of that glucose 6, at the second stage, the glucose 6 will now be converted into fructose 6. So thereafter, if it is converted into fructose 6, it is now going to be converted into fructose 1,6, fructose 6, then 1,6. So that is just going to be what is going to take place. Okay. Now, the next thing now is that will now be divided into two. It's going to be divided into two pathways. The first one is going to be 1,6, that is 3 carbon here, 3 carbon here, and that is where pK is going to act as the trafficator. Do you understand? That is where the pK is going to act as the trafficator. That is, it is going to direct the pathway, whether it is going to go to the left or it's going to go to the right. Okay, so immediately after glycolytic reaction, I told you that the pyruvate acid that will be produced, we have two decisions to make. One of it is that the pathway can go back. It is the organic compound that will serve as the electron acceptor. So it will have the opportunity of either going back into glycolytic pathway, or it will proceed via here. You can see the pyruvate oxidation. So it's going to be directed into the mitochondria, where the whole process of energy generation, which is going to generate more than 32 ATP at the end of the day, which I said it depends on the prevailing environmental condition. Okay. Now, the next one is let's now look at glycolysis. You know, I said glycolysis is a phase that involves spend investment. You will invest. After the investment, you will split, that is, you break down. I told you, after being it has been broken down, it will now enter another stage, and it will pass through this machine. And the other side of this machine, what you will see is 2 molecules of pyruvate I've just told you. Okay, at this level you can see 6 carbon compound. So ATP is going to make sure that 2 ATP molecules will be supplied at this level and at this level. That's why if you remember very well, you will see that between level 1 and level 3, you have 2 molecule 2 times that phosphorylation will take place. What is phosphorylation? Phosphorylation, that is addition of phosphorus. That is phosphorylation. So glucose destabilization via 2 ATP input. So this is the level when the cell will infest 2 ATP, and immediately it has infested 2 ATP, it will now split 6 carbon compound. You can see it here. 6 carbon compound will be splitted into 3 molecules of triols, that is 3 carbon compound. So that is the level of split or cleavage, the level of cleavage, where the 6 carbon compound called glucose will be broken down into 3 carbon triols molecule. So the next phase is the squeeze. you can see that at this level, this machine is going to squeeze the 3 carbon compound, which is triose. All these triose, that is a pyruvate. That is pyruvate. 2 molecules of pyruvate will be produced. So at the end of the day, you should remember something. And how will you be able to remember? Don't forget that I told you at the beginning of this lecture that you don't need to memorize anything. So what you should just do is just remember formula 224. Formula 224. So 2,2 means that 2 ATP is being involved or infested. The second 2 is that there will be 2 trials that will be made. So at the end of the day, you have 4 ATP that will be produced at this level. 4, 1, 2, 3, 4 that will be produced. So when you subtract your 2 ATP that is infested, that was infested originally, when you subtract that 2, at least even if you borrow money to make business, I mean to do any business, you should be able to pay back after you must have made your promise. I mean profits. So the same thing with glycolysis. The cell will not want to hold any ATP, any currency perpetually. It's not going to it's going to pay back whatever. So the energy currency that is starting this business will be infested. That is 2 molecules of that, and one of it will be involved in in cleavage of the 6 carbon compound into 2. That is 3 here, 3 there, and it is that 3 that is being splitted that will now be squeezed, that will be paid off, and that is where you are going to have 2 molecules of pyruvate. So the next one is let's now see the glycolytic ledger and its critical control vessel. I've just told you last week, I think I told you that there is no reaction. There is no reaction that will take place in the cell that the cell will not regulate it. Okay, everything must be regulated, because even if you are making money, and if that money making is not regulated, it's not according to the law, that is telling you that at the end of the day, you'll be talking of recklessness. So that's the same way that glycolytic reaction is going to take place, and it has its own points that it must be constantly regulated. So, and what regulates it is this machine called the PFK1. PFK1, pyruvate kinase 1. Pyruvate kinase 1. So, which is serving as the traffic lights. Serving as the traffic light, you can see anytime there is the traffic light is showing you stop, that is the red light, that is telling you that you already have high ATP. You have in excess high ATP and high T3. That means we have enough energy, so you have to slow down. When you have enough energy, you are already overloaded. That means the vehicle is going to be slowed down. So if this want to pass, the 3 carbon compound want to pass through this, you can see it's coming here. Whenever you see blue, you are referring to 2 pyruvate, molecules of pyruvate. That is the one coming in. If you have this green, that is talking about ATP. And if it is talking about this color, I think this is orange, this is going to be NAD plus that will be talking about. So these 3 will be entering at the same time. But this enzyme called pyruvate kinase 1, that is what is going to happen here. Okay, that means it will be stopped at this level before it will pass through here, and you can still see that at the end of the day, NADH is produced, two molecules of pyruvate produced, as well as two net ATP that is being produced. Now, look at the real scenario. You can see an ATP shortage is not the only problem that can stall the system. It's not when you don't have enough energy. There are some times that NADH plus must not be depleted. If NAD basket runs out, the entire glycolysis assembly line will stop, definitely will stop. That is telling you that the cell must stop and regenerate itself, empty the NADH basket so as to be able to drive the basket to survive. So that is an ATP shortage, which I said is never a problem. Okay, the next one is fermentation. Don't forget what I said. Immediately after pyruvate is being produced, that signals the end of the glycolytic reaction. So the next one is either it's going to go through the journey of Krebs cycle, or it's going to go through the journey of glycolysis again, fermentation, which is going to be a raw material. The NADH that will be produced from glycolysis will now be a raw material for glycolytic reaction. And at this point, where two molecule of pyruvate is being produced, it will now be splitted into two, just like I told you. Okay, lactate fermentation, then this is lactate, this is in animal. So this one talking about pyruvate fermentation, and at the end of the day you talk about ethanol, which is alcoholic fermentation, and that's the point where you will see our yeast being um forming bubbles. So the core insight you need to know is that fermentation saves glycolysis. It cannot outperform respiration. Don't think that fermentation is super, is more super than glycolysis. No, no, because the net yield is still going to remain 2 ATP at the end of the day. Whether you are thinking of um, joining them in the business of cloth selling, or you are talking about shoe selling, if those two, they are going to produce the same, almost in the same range of ROI, that means it doesn't actually mean that it is more special than it. So that's why we cannot consider glycolysis as being special. Don't forget that glycolysis, glyco from glucose, lysis, that is splitting, breaking down of glucose into two molecules of pyruvate acid. Okay, that is that. And pyruvate will act as the ultimate metabolic cross record. So what we are saying is just that the pyruvate, the two molecules of pyruvate that will be produced will now form, will be pivotal. It is going to serve as the engine, the director that is going to direct where the path of the ATP or the whole reaction into success. That is going to be in form of is going to be in form of pyruvate acid, that is fermentation. Okay, or it can proceed via Krebs cycle. So that is the function of oxygen there. So pyruvate pays a toll before entering into the for mitochondrial furnace. Honestly speaking, if you consider a typical cell, be it prokaryotic or eukaryotic cell, if you consider what takes place in them, you will just know that a cell is truly independent. Not only that, a cell is also very, very autonomous. Just like I said, very, very autonomous. They take decision on their own without even thinking of any other thing that can happen otherwise. So what we are saying in essence is in the cell, when you have the pyruvate, look at exactly what happened. This is the three carbon compound called pyruvate. So these two will enter through here, and by the time this one is entering, you just see that in one side NADH will be produced. In the other side, you will see that the CO2 will also be. So it is going to now serve, if it is going to proceed from pyruvate acid, if the next thing is the acetyl-CoA. Acetyl-CoA, that is already telling you that and I remember I used to ask that question, why is acetyl-CoA being regarded as the central or the pivot of cellular respiration? Okay, so pyruvate conversion is the critical checkpoint that commits carbon to the mitochondrial furnace, then generating the first NADH of releasing CO2 as a byproduct. What we are saying in essence is the definition of aerobic respiration will not be completed without you telling them that it is a complete oxidative biochemical processes in which the glucose, via what is called the glycolytic reaction, is going to be sufficiently is going to be um sufficiently it is going to proceed via what is called the Krebs cycle through acetyl-CoA. Acetic cycle is going to produce CO2 and water. Okay, my time is far going. I want you to also understand this is the acetyl-CoA, and when you have the acetyl-CoA, you can see that already it's filling up your basket. There's a basket that is going to be a carrier basket, that is FADH2. Uh, and That one, that is. So if you want to move to Krebs cycle, don't forget that I told you for glycolysis, you talk about 2,2,4. Okay, this one, instead of 2,2,4, you'll be talking about 3,1,1,2, 3,1,1,2, 3,1,1,2. The 3 is for NADH, 1 is for FADH, GTP, ATP, and CO2. So because of our time, I won't be able to actually take my time and explain this the more. But one thing I want you to know is that you don't need to memorize anything. What you just need to know is the right formula to actually be able to get your ATP. ATP is the ultimate. There is nobody in this life that is not existing because the person want to make money. Nobody, nobody. So the same thing, there can never be any kind of focus of the cell or the rationale of the cell if not for ATP generation, money generation. I told you that ATP is the currency of the cell. Okay, without wasting our time, uh, now you can see very well for yourself that these are the functional landmark of regenerating cycle. When you come here at first level, that is CO2 will be SA, you breathe in CO2, then in that CO2 you remove the carbon. Okay, you remove the carbon, and from here through the technical routes there is going to be overload. There is going to be loading of the electron uploaded in form of 3 NADH, 1 FADH2, and the electron carrier load. So thereafter is going to go into where the energy will be offloaded. That is when the GTP token will be released and compatible to ATP. It's just like somebody that has money, but the person's money is in a foreign currency that is not even strong at all. It's not even strong at all. So that is the same way that you can think of GTP and ATP. ATP is the universal currency of the cell, but there are some times that if ATP is not available or in readily supply quantity, that means they can also make use of the lesser one, that is GTP. So you don't need to memorize 8 steps, just understand the continuous generation of oxaloacetate. Okay, now another very important thing that you need to know is that this pathway dey also crosstalk, dey communicate each other. Do you need energy? At what point are you? How much do you need? Okay, when the furnace is full, that is, you've already gotten enough energy. When it is full, the next thing is that the cell will stop adding the more. So the cell automatically will shut down because it already has more than enough. It is only somebody who is not even serious at all that even when you must have eaten to repletion, you must have eaten to satisfaction. So the next thing is that you are still consuming more food. At the end of the day, even the small one you have eaten, you are going to vomit it at the end of the day. So what we are saying is that these pathways, they also crosstalk. They talk. Do you need more? Don't you need? Are you satisfied? Are you repleted? Do you need water? Do you need? So that is one of the essence or one of the reasons why and where you have our cell or the cell, any cell at all, be it prokaryotic, eukaryotic, any cell at all, they do what? They crosstalk each other. So PFK1, you know, this is a glycolytic pathway, which is the originator or the first. Okay. Thereafter, you have the glycolytic pathway. Then you have Krebs cycle in form of this. So that is the metabolic pathway to show that cells do interact. Okay, they interact, they communicate with each other. They know when their own colleague is getting uh, more energy, or when their colleagues is already depleted, is already satisfied, so that they won't continue to generate more energy even when they have more than enough. And the same way, if they are not even if they are in short supply, they will still communicate with each other so that they will be able to know the next thing that will be done. And if you now look at this table very, very well, this is a comparative table that compare glycolysis, fermentation, and Krebs cycle. I told you in form of location, glycolysis is located in the cytosol, and the fermentation also located in cytosol, and Krebs cycle in mitochondria. And what is their main purpose? Split glucose, regenerate NADH, that is fermentation, and extract electron. So I want you to study this table so that you will be able to know exactly what. So the master audit refill latent power, you need to understand this. I think it is something you can on your own DIY, on your own do it yourself. So take away the vast majority of glucose energy is not in ATP yet. It is sitting as latent power in these 12 loaded electron carriers. Okay, anyway, don't let me bother you about that, because when we are running to solve a problem, we are trying to solve the problem of students not memorizing, because you will just end up, even if you memorize, you might even end up not even remembering it. Any day or any time you might even need it. So, but if you know the basics, if you know the rudiments, if you know the beginning, how to combine them, how they come together, so honestly speaking, you won't in any way uh, regret. Okay, debugging major misconception about the operating system, that is the myth surrounding fermentation and ATP. Some people believe that fermentation makes a lot of ATP. No, it makes zero. Extra ATP is only purpose to generate NADH, so glycolysis can continue. So that is that. The myths surrounding the Krebs cycle and ATP. People believe that Krebs cycle is an ATP factory, whereas it is an electron harvesting machine. Most ATP is produced later by what is called oxidative phosphorylation using the Krebs electrons. So what I'm trying to tell you is that when you get home, you can um, see this table and be able to talk about some of the misconceptions that is surrounding all this. So uh, without wasting our time, I want to really salute your listening attention tonight, and I wish that we ended earlier. So with this, let me just display just 5 questions instead of 15. Let me just display 5 questions for you, and that is going to be your post field. Don't forget that this is Edu Interactive High, where we democratize learning, where we want to bring about high tech, educational content to you, okay, free of charge. Free of charge. So we have democratized, because you can see you are already at the four corner of your room and you are listening to this. Then apart from that, if you visit our website, you have the AI already that is waiting for you to ask it question, any question at all. So, and that is the reason why that today, that's the reason why that even if that one will teach you, I cannot teach you 24/7, but the chatbot in our website can teach you anything for 24/7, round the clock. Ask it any question. When it was launched, I just told you, I just asked you to go and check whether it is functioning or not, and people have tested it and discover that it is perfectly working. Another thing that Edu Interactive High has been able to do successfully is to be able to integrate our platform, is be able to integrate the website. um we have integrated it with Claude AI. That is telling you that the same way when you prompt the AI in Claude is the same way that the website is also going to respond to you, and it is this chatbot that is going to help you to choose depending on your level or status in terms of knowledge, that will be so fit for your level, be so fit for your age, that will be so fit for your technical needs. So, and that's why you should see this chatbot in our website as a learning assistant for you, a learning tutor that can teach you anything related to biology, anything related to chemistry, anything related to um, physics, anything related to medicine, anything related to genetics, bioinformatics, anything related to microbiology at the end of the day. So what I'm trying to tell you is just that you should feel free to be able to meet the chatbot, ask it any question, it's going to teach you. I just told you sometimes ago that very, very soon is because of my engagement these days. I will have also installed the widget that is the chatbot that will be able to also cross talk with you. The chatbot that will also enable you to do a lot of things, uh, master a lot of things that sometimes just prompts the chatbot. I want you to teach like Mr. So and so. He's going to pick, he's going to search through the net, look for any content that is already linked with him or her. And the next thing you will see is so what I'm trying to tell you is that before we go, let us, I want you to take this journey of the metabolism quiz so that you'll be able to know whether you have really learned or not. So please, this is going to be just 5 minutes. I mean, it's going to be five questions. Okay. According to the simple acts toolkit, which term describe the summary of what goes in, what comes out, and where energy or electron move within a pathway? Okay, so is it called the control point? Is it called the story? Is it called the investment phase or the ledger phase? Okay, that's a question one. I'm only going to display only five because of our time. Okay, so the next one is this. What is the net yield of ATP and NADH per molecule of glucose during glycolysis? So you should be able to pick one out of that option. Okay. So the next one is question 3. Question 3. So we have two more questions to go. Question 3. Two more questions to go. Okay. Oh, somebody is saying we should go to, I should display the rest to make it 10. Somebody is saying that. Okay, if that is what you want, let's make it 10 because initially it was meant to be 15. First test and second test. And the first test is going to carry uh, 15, whereas the last one I said it's also going to be like going to be initially 5, but now people are already commenting that it should be more than that. Okay, which I quite agree with them. Okay, this is question 6. So we only go as far as 10. Only 10 questions will be entertained. Okay. So we have one more question to go. So don't forget that tomorrow, by this time tomorrow, that's 8 o'clock, we are going to have toxicology. That's for environmental.